Hi everyone, how's it going? My name is Ramon, and today I'll be taking a look at the CRISPR-Cas9 complex and seeing why we should care about it. So first off, what is CRISPR-Cas9? Well, CRISPR is short for Clustered Regularly Interspaced Palindromic Repeats, which is a long and intimidating name for a section of DNA that is essentially a kind of bacterial immune system. It works by slicing the sections of foreign DNA, like from viruses, to render it inert.
Alright, cool. So
that's CRISPR. What about Cas9? CRISPR functions by using specific proteins known as Cas proteins to do its dirty work.
Cas is one of the
best Cases at its job.
There are a few other names
we need to go over to better understand the whole mechanism of how this thing cuts DNA.
For one,
CRISPR-Cas uses Cas9 like a scalpel,
but it needs to
know specifically where on the DNA strand to slice, which is where RNAs come in. CRRNA acts like a targeting system for Cas9, while tracer RNA recruits Cas9 for the job.
These are both
naturally occurring RNAs, but scientists have also come up with CGRNA, which does the jobs of both of them at the same time. Neat, right?
Alright, so we're almost done. We have the mastermind
CRISPR, we have the recruiter and targeting system RNAs, and we have the slicer Cas9. But Cas9 can't just land on any old DNA section. It needs to land next to a specific combination known as PAM.
Even if we have all
the other members of the team, without PAM to guide Cas9 in, none of it will work.
So that's all
really neat, but why does it matter to us that a bacteria could slice open a virus?
Well, scientists have learned how to use
CRISPR-Cas9 as a gene editing tool. Scientists have been able to introduce cancer into model organisms, which actually sounds kind of mean, but really helps us understand cancer better and how to fight it.
Also, thanks to the complex,
we have a much better understanding of the human genome and what specifically happens when you turn on or off a specific gene.
We also have the
possibility to create better crops, with none or less of the side effects that come from current GMO strands. In the area of medicine, scientists in 2013 managed to restore the defective gene responsible for cataracts in mice, restoring their eyesight.
If we could transfer
that technique over to humans, we might be able to cure cataracts.
In the same year,
2013, another scientist was able to restore function to the gene responsible for cystic fibrosis in an extracted cell.
3 years later,
another one did the same for the gene that causes a type of muscular dystrophy.
So, I know what you're thinking.
Gee, Ramon, that sounds great. With this kind of tool, we could fix all sorts of genetic problems.
And
he might be right.
But there is a
potential downside to this.
For one thing,
doctors and scientists do make mistakes.
When
dealing with genetics, it only takes one to create a cascade of negative effects. Take cystic fibrosis, for example. One defective gene causes a lifelong illness. If we're not careful around this tool, we could create all kinds of man-made genetic diseases.
The government?
That's not the only bad news. If we had the power to cure genetic deficiencies, what would stop us from creating genetic enhancements? Those of you who read the Divergent series might know what a problem it would be if people were able to discriminate against each other on the basis of genetic purity or inferiority. And even if that didn't happen, who would get to decide who gets genetically enhanced or even cured of genetic diseases? The government? Yeah, that would be a great idea. We know that you can always trust the government. Now, you might think this is all a bunch of hullabaloo or science fiction, or you might think that these issues are far in the future, not our problem. Well, let me introduce you to Dr. He Jiankui, a Chinese scientist who was arrested in 2019 for illegally and unethically using IVS and CRISPR-Cas to edit the genes of 3 unborn children.
This isn't some
far-off future problem. This affects us now.
Now, I'm not trying to be an
alarmist or a conspiracy theorist or whatever, but it's important to understand the gravity of a tool like CRISPR-Cas9 and what it can be used to do.
So, in conclusion,
CRISPR-Cas9 is a powerful, cutting-edge genetic tool with the potential to do a lot of good or a lot of bad.
Which will it be?
Which will it be?
I guess that'll be up to us.